Good afternoon, everybody. I was asked to say a couple of words about the issues of traceability and laboratory testing, and it's always interesting. I think the organizers have really strived for introducing diversity here, because here is a representative of probably the biggest agricultural state, and then they follow up with probably one of the tiniest agricultural states in the country. If you look at this picture, I apologize, it's a little bit blurry. This is a picture that was taken on a cloudy afternoon in Nevada last week. And obviously, you do realize that that's a lie, because we don't have cloudy afternoons in Nevada. This is actually a very famous picture. This was taken in 1896 in the Eastern Transvaal in today's South Africa. And this is 1896 is when the rinderpest epidemic actually reached South Africa. It probably was introduced onto the African continent in 1893 into Ethiopia, and then spread south for the next 3 years, probably eradicating 95% of all domestic cattle owned by both European settlers and indigenous peoples in this area, plus about 95% of ungulate wildlife in some of these areas. Subsequent to that massive loss of wildlife, obviously there was also loss of predator species. The entire ecosystem in areas probably pretty much collapsed. Now, one of the positive effects of this massive livestock disease that also brought famine to essentially half of the continent was the eradication of tsetse flies south of the Limpopo River, and the concurrent eradication of sleeping sickness both in domestic livestock and in humans. So, let's look at the glass as if it were half full. The massive death loss actually brings health for everybody who survives. However, on a political basis, the assumption is that this kind of animal disease probably pretty much set the path for how Southern Africa developed politically, because of the complete impoverishment of all the native peoples that were subsequently, or that previously had dependent on cattle agriculture. It also led to the founding of the Onderstepoort Veterinary Institute by a Swiss veterinarian, Sir Arnold Tyler. And I don't want to belabor the fact of rinderpest too much, but just to make my point here, we probably had rinderpest for 2,000 years in Europe. We had epidemics every 40 to 50 years. The human losses during those 2 millennia are untallied. And the assumption is, and this is all, feeding into the One Health concept here, that this morbillivirus probably was an ancestor or a contributor genetically both to the measles and the canine distemper viruses. Also led to the establishment of the first veterinary college in Lyon, France in 1750, and then subsequently throughout Europe to the establishment of basically all the other veterinary colleges within the next 150 years. And that for the first time was where animal disease actually changed government to a very significant point where it became institutionalized, because this was an act of 18th century emergency management to basically try and get a handle on this nasty disease that really held back the development of entire nations. If we look at our vulnerability in terms of infectious and emerging diseases, and we in the diagnostic labs basically always get to play with them, if not first, then certainly second. Obviously, the most publicized disease over the last decade or so was the introduction of West Nile virus into the United States, and that is a vector-borne disease, which makes it even a little bit more complicated in terms of traceability. The diagnostic assays were available within 12 months. They were developed by veterinarians, just like the disease itself originally was identified by a veterinarian, Dr. McNamara at the Bronx Zoo. And this was one of those occasions where actually the different branches that should have been involved in the control of this disease got together fairly quickly, and we all got on the same page. We participated and collaborated in surveillance and control using a multi-pronged approach, different types of testing, avian testing, predominantly indicator species, equine testing, and equines were great. Tracer, essentially for infection transmission until the spread of vaccination. We still test every year about 3,000 mosquito pools in the state. The only data point that was collected, surely, that didn't contribute to actually disease abatement was human disease data, because that is protected. So in terms of protecting human health, the only data that isn't available is human disease data. So vector control agencies very heavily had to rely on all the information that was gathered together with the this biological specimen of avian, equine, and mosquito origin, because they all came with a longitude and a latitude, and every single submission was clearly identified as to origin. On the downside, however, we were just told that the 2011 presidential budget, all vector-borne disease funding has been stricken. So this is obviously not a good sign in terms of has Washington or have our lawmakers heard the message of what is required to actually get a handle on emerging infectious diseases. So one of the other issues that I would like to point out to you, you in the Western United States are probably familiar with this issue right now. The spread or the risk of spread of what I refer to as the golf course rabies in the southwestern United States. A big brown bat rabies strain has adapted from foxes to skunks on skunks to golf courses on golf courses in Flagstaff, Arizona. This is, by wildlife officials, this has been termed an environmental disease. Again, this is something that humans have caused. Because there's usually not a huge population density of skunks anywhere in the desert southwest, because there simply isn't enough biomass to sustain those. On golf courses, which are obviously a very artificial environment, that, however, has become an opportunity. There's a lot of open water, there are a lot of mosquitoes, hence there are a lot of bats. There are also a lot of skunks. So this is where probably this transmission and adaptation of a previously bat-adapted strain has now happened and is now a new strain in terrestrial rabies. And this goes fairly quickly. So a lot of us that are under 50 are probably going to see several of these cases before we retire, if we ever get to retire, before we get there. Because this type of very complex biological adaptation of a pathogen to a new host doesn't take very long. It takes years, sometimes a decade. It can take a lot less. It can take a couple of weeks or months depending on what host and what agent you put together. So the first detection in a non-bat for this rabies virus was in 2001. By 2004, mere 3 years later, there was a significant increase in this particular strain in terrestrial rabies. By 2008, this was considered to be completely adapted to these new terrestrial mammals, and we faced a new problem, or rather we face an old problem now with multiple problems, and that is, how do we control this disease? Skunks are very hard to vaccinate because you can't give them oral bait vaccination, so you essentially have to trap them, vaccinate them, and release them. Otherwise, there were huge outbreaks from the neighborhoods where they found positive animals that they did not want a skunk eradication program. And obviously, as in each of the disease outbreak cases, data, origin or location data is diligently mapped, and even if you have that data, eradication is always a very costly proposal, and the tools in this instance are not fully efficacious. If we look at wildlife disease investigations, which our lab also participates in on a regular basis, right now a lot of states have concurrent outbreaks of Bighorn sheep pneumonia in the western United States. Washington, Montana, Utah, and Nevada have some very severe die-offs this winter. And our colleagues in the Department of Wildlife, what do? They do aerial surveillance, and they collect data, longitudes and latitudes for everything they do. For aerial surveillance, if we sacrifice individual animals for diagnostic workup, if we take live samples and treat these animals, obviously we identify location data and we individually identify these animals. And this is from a psychological standpoint, I think this is a very interesting phenomenon from my perspective. It seems that people who don't easily have access to the animals that they manage, if they actually can tag an animal, it's like a badge of honor, it's like a milestone of accomplishment, how many animals you can tag and individually identify in your career as a wildlife manager. for us in agriculture, where we have easy access to animals and it would be just a breeze to identify them, there seems to be a somewhat different mindset. So I'm sure you've all read about the court case of the Amish farmer who refused to participate in mandatory animal ID. And just like Dr. Breitmeyer, I've chosen the Scrapie program to actually illustrate that individual ID for animals that enter the marketplace is actually, in one industry, is very highly accepted. It was relatively easy to convince people that there was a benefit not only for the regulatory veterinarian, but also for the person, for the producer, and essentially for the national flock. If we ever have to test in a lab for a high-impact disease with a large number of affected animals, obviously there are two bottlenecks. There's, first of all, the testing capacity. But before we even get to the testing, how are we accurately capturing individual IDs? The way we're doing it right now for a lot of animals, and all the problems with capturing individual ID manually have already been pointed out, that is an enormous issue. From my perspective, I don't think that can be overemphasized. The lack of permanent individual ID hampers any disease control program, whether that is a fast-spreading or a slow-spreading disease, and particularly for breeding animals, which are high cost and high impact. A lack of individual ID puts us in a very difficult position on a number of occasions. Same with lack of premise ID. And I don't want to, use another federal program to illustrate the point. But for us right now, the biggest issue that we have in terms of traceback is the lack of identification of both premises and individual animals in T. fetus epi investigations. And for us in the western United States, trichomonas fetus is a very significant parasitic disease in cattle, and it absorbs a vast amount of personnel time right now. And when we do epi investigations, we run into problems that could be very easily prevented, but which currently we have to deal with. We've run into occasions where during epi investigations and by reviewing our lab records, we've run into a bull that belonged to Joe Smith, or a contact that was J. Lazy S, or a submission from a veterinarian on behalf of Shelley and Joe Smith, or J. and S. Smith by another veterinarian. And for us to be able to make the connection that all of these submissions on all of this data that is strewn around in different databases actually belongs to one and the same premise and one and the same herd is incredibly time-consuming and is very laborious. And my question is, T. fetus is not a fast-spreading, high-impact disease by any stretch of the imagination, and it's so hard to get a handle on that under the condition that a lot of people ranch and manage in the United States, particularly in the West, on public lands. What are we going to do in terms of traceback if we do have a high-impact, highly contagious, fast-spreading disease? Data management, as Dr. Breitmeyer has pointed out several times, is a crucial issue. And when we deal with submissions and we process our samples in the lab, we deal with program managers that have their own database where they keep data on the livestock populations, the wildlife populations, or whatever population that they're managing. And then we get part of that information. We do that with the Department of Wildlife, or in Nevada with the Department of Health and Human Services, who have deferred the arbovirus testing to our lab. We have to enter all this data into our lab information management system. All our West Nile virus testing we have to separately enter into ArboNet. And then again, for our avian influenza testing, we have to enter the data again in a USDA database. So from our perspective, that is very, very time consuming. It's very labor intensive. And from a data mining standpoint, this is obviously a huge disadvantage because there's all that data that really is only accessible by one or two individuals, whereas if it were accessible through a combined database to a lot of people, that could really be mined, which right now we will never be able to reach the full potential of the data we already harvest and generate. So what is next, and how do we prioritize where we put the resources? Obviously, part of the One Health concept is the realization, or the origin of the One Health concept is the realization that we probably have only seen the tip of the iceberg at this point. This year, well, in 2010 so far, there have been dozens of papers published of potential new and emerging and mutating infectious diseases. One of the issues that I just found in a paper, Noro and sapo-viruses, each of them occur in humans and in different livestock species. The question is, are they transmissible back and forth? And how tight is a species barrier if there is a species barrier? Does that have consequences for food safety, particularly in the case of Noro-virus, which is a food safety issue at this point? Or is it just occupational health for consideration for the workers on the farm? Pigs are now turning out to be a potential hepatitis E reservoir. H1N1, there is a definitive link between humans and pigs, obviously. And when do these diseases become a concern? Who determines that? Right now, is it Katie Couric who determines that? Is it anybody else in the media? Or are we the ones who determine, based on the damage that can be done, whether this is a problem or not? And a very important problem is to determine whether these are newly detected disease agents or whether these are agents that have been around all the time, but they've just escaped notice because at some point they haven't actually caused any disease, or can they mutate into anything? So these are the questions that diagnosticians have, and we usually, pose them to our regulatory veterinarians. Just like if we do have a result that is of consequence in a regulatory or in a public health arena, it's usually the public health or the regulatory veterinarian that actually has to deal with it. My biggest concern is, if we ever do get it, will we see it? And when we see it, will we know what we're looking at? One of the big issues that we have right now is that we've abandoned those so-called discovery diagnostics like virus isolation and electron microscopy. Because if you put a virology sample under an electron microscope, you might be looking at what you were looking for in the first place. You actually might find that, but you also might find a whole lot of other things that you then can follow up on should that be required. Right now, if we test a swab for avian influenza or exotic Newcastle disease, the only thing that we're looking for is that one or maybe those two diseases, and whatever else is in there and might cause disease, we don't have the foggiest idea. And it will escape us. It probably does all the time. In veterinary diagnostics, microarray analysis, like in human disease, like they're trying to push human disease investigations to, are going to be light years away because of the cost issue. And according to many of the papers that are published in the CDC journals, is we're one antibiotic or one antimicrobial away from the next plague. So the human-animal bond is basically what got us here in civilization, and it's always been very tight. We're basically always taking the blame in livestock industry on how we are causing disease as opposed on how we are feeding the world. So if all of our friends who think that we should go back to a very natural lifestyle where we should just, gather and hunt, there are several very compelling papers out there that basically make the connection between simian immunodeficiency virus and human immunodeficiency virus, and how that virus seems to have jumped the species barriers through the consumption of bushmeat. So to assume that the consumption of wildlife is necessarily wholesome and lacks any risk is bizarre. But it is propagated because this is right now, it's fashionable to have that impression, and we're doing very little to disperse that because we're all very PC. If we look at the diseases that are transmitted, currently in our regulatory importance, so who was there first? If you look at the genetic relationship between Mycobacterium tuberculosis and Mycobacterium bovis, they are so closely related that at this point it's a chicken or the egg question. One seems to be the ancestor of the other. We don't know who was what. Did we get it 10,000 years ago from a Bos taurus species because we used to live in the same cave with them because they warmed us, and so we kept close to them in the winters? Or was it a Bos indicus cow somewhere in the Indian subcontinent that gave it, to its herder or to the person who was pushing the plow behind it? Or did we give it to them? That is unclear at this point. Not that it makes much of a difference at this point, but these are all considerations that we need to look at. The other part of the human-animal bond is companion animals. If you look around how many MRSA strains are isolated from skin lesions on cats and dogs, you would be surprised. And this human-animal bond is probably going to get a lot closer as we make giant strides in biomedical research. We're probably about 10 years away from xenotransplantation. All those people that are currently waiting for kidneys and livers and pancreases, 10 years from now, you can probably have it custom grown out of your stem cell in a pig. But what are the consequences of that? Right now, we already know that there are, porcine endogenous retroviruses that actually, if you co-culture them if you culture pig cells and human cells together, they make a live retrovirus just by mixing two genomes. All of our vectors that we use to actually introduce genes as part of gene therapy into a genetically ill person, right now we're checking them against the human database to see if there's overlap and can they actually make a infectious virus again. But there are 6.5 billion human genomes out there. Are we going to basically drop this great opportunity to help people just because there is the next disease around the corner that we might create through medical intervention? These are all things that have to be considered. And then once it happens, once the next big disease is here, what are we going to do? Communication. That's a Sisyphean task at best. Most of the time, I think we find ourselves in the situation where we do damage control. And the first reaction is usually shoot the messenger. But the very first question that we're going to have to answer, no matter what we find, is where did it come from? And right now, I'm afraid, most of the time the answer is going to be, I really don't know. And also, there's probably not a lot we can do to find out where it came from. So there's multiple issues at that point where we do find a disease. And one of the things we realized in Nevada in 2003 when we had an END outbreak in Las Vegas, and we're not even a commercial poultry producer, but we are a state that lives off of tourism. So we had to post these signs that Clark County, Las Vegas was under quarantine, but at that time of the year, December, January, February, we get all of these retired folks that come through with an RV and their parrot. So our governor did not want us to post any signs anywhere that says, if you got a parrot, you got to go through Arizona. You're not coming through here. There's not going to be a lot of support because it's shooting the messenger. You have to tell people something that people don't want to hear, particularly people in power, because it costs money to deal with it, and they don't want to hear that. And up front, we very rarely take the opportunity to actually get out a positive message. We are, I think, the product of a frugal industry, and the industry has raised frugal professionals. So a lot of the dire financial consequences that we always complain about, we've probably caused ourselves because we've never been able to explain how what we do is worth a lot more money than we actually get. So I tried to figure out what it is, what we cost. I can tell you right now that the Division of Animal Industry in Nevada costs a household $1.11 per year. $1.11. I could not figure out what the State Health Laboratory actually receives in terms of general fund money, because it has so many different account numbers where it gets money from so many different programs that it's impossible to figure this out. And several of the slides yesterday showed the same disparity between NIH and ARS funding. So, not to belabor the point, but at this point my question is it going to take this to get an animal ID system in place? I certainly hope not. And the rhetorical question at this point is, why do we do all this? And I guess this is my last statement of the obvious.